Cellular structures part 2. Hopefully we'll have fewer cat interruptions this time. I just jinxed myself. Yes, hello. I did mention you. I did. Hi. All right, so there are lots of other organelles in the cell that are not part of the endomembrane system, remembering that the endomembrane system is the ER, the vesicles, the Golgi apparatus, and then those secretory vesicles or the lysosomes. So one of them, these are peroxisomes, so mentioned before that these are the things that are going to process hydrogen peroxide. So as we start metabolizing, our bodies take diatomic oxygen, O2, and break it into monoatomic oxygen in order to generate energy. Free monoatomic oxygen is going to have a free valence electron. That makes it a free radical. That makes it super reactive, makes it super dangerous. So the oxygen free radicals that are not immediately consumed in the process of cell respiration can do cellular damage. So it is the job of the peroxisomes to take that, combine it with water, and make hydrogen peroxide. Hydrogen peroxide is less dangerous than monoatomic oxygen. However, hydrogen peroxide is also very unstable and very dangerous. So what the peroxisome does is it makes the peroxide, then it contains this enzyme catalase or peroxidase, and it breaks that down into water and diatomic oxygen, which are both safe. So it takes something really harmful, makes it into something less harmful, and then makes it into something safe. So these also play a role in detoxifying alcohol and processing the fatty acids from lipids for energy. So they are important. There are also a special type called glaucosomes in plant seedlings for breaking down the fats in seeds so that seedlings can. We also have some double membrane organelles, so that means they have their normal membrane, then they've got another membrane around them, so that makes them double. These are the chloroplasts and the mitochondria. What you need to know about them right now, essentially, mitochondria make ATP, yes, the powerhouse of the cell, and chloroplasts do photosynthesis. The question, though, is how did they come about to have a double membrane in the first place? And this goes to the whole question of eukaryotic evolution. So how do we go from a prokaryote that doesn't have a nucleus to something that is a eukaryote with a nucleus and also have these extra organelles that help them do much more important things? So the thought and the theory is that the nuclear membrane resulted from the cell membrane getting too big and kind of folding in, and then it kind of wrapped around the DNA and then formed the membrane, and that made it specialized and better at doing its job. So therefore it had an advantage, and so that process continued on. There is a video in the playlist about the endosymbiotic theory. It's a TED Ed. It's not a TED Ed talk. It's a TED Ed education video. So I suggest you watch that. But the other part of it, how do we get mitochondria and chloroplast? The thought is that you had a heterotrophic cell, something that wasn't able to produce its own energy, engulfed another prokaryote. Therefore, you got the second membrane around it. But that second organelle, that second prokaryote, started producing more energy. That's the first mitochondria. And that gave an advantage to that cell, gave more energy to do stuff. And so they formed this mutualism. And then the same thing happened with the chloroplast. The chloroplast used to be a free living prokaryote. It got engulfed, and then it started helping that cell become a photoautotroph. It is suspected that the chloroplast happened later, and the reason for that is the both the mitochondria and the chloroplast have their own DNA, but some of the DNA that the mitochondria need have been incorporated into the nucleus, so they think some gene transfer may have happened, whereas the chloroplasts are completely self-sustaining. Okay, so this picture. Here is your ancestral prokaryote, and here you can see kind of these infoldings of the cell membrane, which eventually form around the nucleus and then form the endoplasmic reticulum. They get into having specialized functions, which gives a selective advantage. Then you have the incorporation of the mitochondria. We get this engulfing of this free-living prokaryote. It becomes the mitochondria, and then we diverge into our heterotrophs, which only have mitochondria. And then later we have the engulfing of the free-living photosynthetic prokaryote, and that becomes the chloroplast. And then you've got your lines, which evolve into your fungi and your plant-like protists, not your plant-like protists, your animal-like protists and your animals. And then you have your line that develops into your plant-like protists, algae, and your plants. Now, what is the evidence for this? Both have their own DNA. It is circular, just like prokaryotic DNA, and there are no organizing histones. That's the stuff that wraps up the DNA and keeps it nice and organized. They have that double membrane, so as the cell engulfed it, it wrapped the membrane, giving it two. They are the same size as modern prokaryotes, and they have similar enzymes and membranes as the prokaryotes. Chloroplasts make all their own enzymes. Mitochondria don't, so that means that's why they think it was a much earlier engulfing of that mitochondrial ancestor. All right, what do you need to know about mitochondria? Cell respiration, makes ATP. We'll talk about all of this stuff in great detail later. It's in almost all eukaryotes. There is one group, the Giardia, that don't have mitochondria, and they just exist off of anaerobic respiration. These are all the structure in the mitochondria and all the parts. You don't need to know that now. Chloroplast, site of photosynthesis because of chlorophyll, parts, structure, etc. We will learn that later too. All right, the cytoskeleton. This is the inner working of the cell. It's a bunch of fibers and tubes, has lots and lots of functions. One of the main functions of the cytoskeleton is to give the actual internal framework of the cell that gives it its shape. So muscle cells are long and stringy, nerve cells look like trees. So they have these specific shapes, and they are given that specific shape because of the cytoskeleton. It is the framework. It's like the internal support of a house that gives it its shape. That is the cytoskeleton. Cytoskeleton also holds organelles in place. They don't always just free float, and that helps give some order and structure inside of the cell. Other things, cytoskeleton is used for movement. We have cilia and flagella on the outside of the cell that wag and wiggle to move things. You also have the internal movement that causes contraction. It is the cytoskeleton that's responsible for muscle movement. We're going to talk about muscle contraction later. And also those vesicles that we talked about moving from the endomembrane in throughout the endomembrane system, they don't just kind of freely float. They actually move on a monorail system using some proteins, which is super cool. And there's a video in the playlist that shows that, and it's super awesome. It's one of my favorite videos. And then as these cells manipulate and change the cytoskeleton, it can cause the shape of the cell to actually move and change. The cytoskeleton is actually kind of a pressure sensor as well. So when we press on our fingers and we feel that, it is because of the compression of the cytoskeleton, and that triggers the pressure sensors in our nerve, those pressure sensing nerves to cause them to send a signal to our brain. It can also stimulate other cell processes, moving organelles, so it's just kind of like a pressure sensor inside the cell. So this is our organelle monorail. So this is the vesicle. It's attached to these proteins. The ATP causes these to move like little feet, kind of like drags it along the cytoskeleton, this one being in the microtubules. The video of it is really cool. 3 basic types of fibers in the cytoskeleton. There are microtubules, which are tiny tubes. You can see in this picture, it's a corkscrew. Actin filaments, and then intermediate filaments. Microtubules are actually the biggest of these. Actin filaments are the smallest, and intermediate filaments are your Goldilockses. These are all some information which we'll be going over in the next slides. These are the 3 components of the cytoskeleton compared. So microtubules are the biggest, microfilaments are the smallest, intermediates are in the middle. These are hollow tubes. These are both solid, but these are really thin, and these are really complexly formed, but they're super tough, and that's important for their function. And the proteins that make them up, tubulin for microtubules, actin, and then keratin. If you have heard of keratin, that's what is the main component of our skin. It is also what it makes up hair and fingernails and feathers and scales. So that's the stuff that is our basic exterior for all of the higher vertebrates. All right, microtubules have several functions. They give shape and support to the cells. One of their main functions is going to be the spindle fibers to separate the chromosomes in mitosis and meiosis. So when everything lines up in the middle and we have to separate the chromosomes into the new cells, the spindle fibers are what attach to the center part of it and help separate them. They make up centrioles, which are these things that the spindle fibers grow out of in animal cells, but nothing else. But we'll talk about those later and how they're not really that important and why are they there in the first place. They make up cilia and flagella for movement. And then, of course, they are that internal rail system to move vesicles around inside the cell. The anatomy of microtubules, they're just a bunch of little tubes put together inside of a long bit. You do not have to know all the internal structure. The only thing that I want you to take away from this is that these tiny little things are super, super detailed, and it's kind of super cool. So this is how they look. In order to understand the way cilia and flagella move, you do need to know that it's all of these tubes, and then in the tubes there are these tiny little protein molecules here, the things that look like little boots. They're called dynin molecules, and they help move the tubes back and forth to make the cilia and flagella waggle. So cilia and flagella, we tend to think of them as moving cells around. So the little things in pond water, flagella are on the ends of sperm to make them do their thing. But a lot of tissues have cilia on the surface, like the lining of our trachea and on the inside of our lungs. There's lots of cilia to move mucus and dirt and dust and spores and bacteria out of our lungs. And then we cough that up throughout the day and swallow it into our digestive system to die. There's cilia inside of our kidneys to help move things, and so it's not just for movement of cells, it's movement of materials across cells. Cilia tend to be numerous, flagella 1 to 8 depending on the cell. There is a video of cilia movement in the playlist, which you can click on and watch if you want to. The way that they work, cilia, they're like oars on a rowboat, a crew team. They kind of have that power stroke and then the recovery stroke. And the thing that is amazing about cilia on a single-celled organism is that they are coordinated. They move together because you think about a crew team. If they weren't, listening to the guy telling them when to stroke, they'd just keep clacking their oars together and it would be a mess. That happens in these single-celled organisms as well. There's no nervous system. There's no brain in that cell. So there is some kind of chemical communication going on inside of that single cell to coordinate that movement. And that's amazing. And scientists aren't sure what's happening there. Flagella is a whip-like structure that kind of goes back and forth, kind of like a snake moving across the water. Basically, you've got these microtubules together, and then the dynein molecules grab them and cause them to slide. And then on one side they're moving, and then it shifts, and then it moves on the other side. And so as they are moving back and forth, that's what causes the waggling movement. And that's really all you need to know about that. There are some complicated videos online if you want to look those up. But you can see how molecules kind of grab it and move it. This is also basically the same process of how muscle contraction works, which we will get into in depth. It is one molecule grabbing another and sliding it past itself. But what happens is it just kind of goes back and forth, and that's what causes it to move. Sperm, important, reproduction, babies, etc. And so here's a kind of a cool picture of a paramecium and all the cilia on its surface. So this is the power stroke, and this is the recovery stroke. So it's like boom, and then it kind of backs up and boom, but it's all cool. So all of those things are coordinated. Otherwise, if it was all random, it just wouldn't go anywhere. Moving on from the microtubules to the smallest of the elements, which are the microfilaments. These, again, are made up of actin. And one of the things that they can do is they can actually help make the cytosol or the cytoplasm thicker. So it's just the same way that gelatin works to make water into jello. So if this is your water, when you put in gelatin, gelatin is a protein that is collagen that comes from the skin of horses and cows and grinding up their hooves. And when you put it in water, it begins to expand and it starts to cross-link like this. As it cross-links, it impedes the ability of the water to move, so it becomes more firm and more solid-like, even though it's not a true solid. It's a semi-solid. And that's what the actin filaments can do inside of the cell. They can help solidify and make the cell firmer, or those filaments can break down, allowing the cytoplasm to flow more. Microfilaments are also very critical in the process of muscle contraction. This is what makes up the contractile fibers in the muscles. So here you have an actin filament, and you can see that it is made up of lots of little actin subunits bound together. And what happens in muscle contraction, you have two proteins. You have the actin filaments, then you have a thicker, bigger protein that is called myosin. And together, these work to pull the muscle cells shorter, and that's the basic process of contraction. So inside of the muscle cell, you have the unit that is known as the sarcomere. And inside the sarcomere, you have lots and lots of the actin filaments right here. And then the purple ones are the myosin. They're not really purple, but just for ease of viewing, that's how they're often colored. So what's going to happen, the myosin has these little heads right here, and those are going to grab the actin and pull the actin together. So imagine that they're like a little crew team, and the myosin heads are the paddles, and they grab the actin and they pull them together. They don't all do it simultaneously, but that's the same. That's the basic idea. In the muscle, so if you were looking at your muscle, that is made up of different muscle fibers. So if you've ever torn apart a chicken tender and you see those strings of protein, those are the muscle fibers. And those muscle fibers are made up of myofibrils, which are then made up of the sarcomeres. You don't really need to know that. That's just the general structure. Wrapped around the sarcomere is some smooth endoplasmic reticulum, which is now going to be called the sarcoplasmic reticulum, which is made up of the actin and myosin. And these have all these different labels, the middle line or the M line, the I band or the light band because it's just actin, and the dark band because it's the actin and the myosin. You don't need to know that. All right, so this is your basic structure. So here's your muscle. It's attached to a bone by a tendon, and then that is broken down into fibers, and then you've got your muscle cells, and then it breaks down. One thing that's interesting about muscle cells is that they're very, very large and that they have multiple nuclei throughout each cell because the nucleus has to be coding for RNA to make new muscle proteins. And if you just had one nucleus for that very, very large cell, it couldn't make enough RNA for that process to happen. So it's kind of like having multiple brains inside of a body. All right, so the muscle contraction process is known as the sliding filament model. So basically you think, oh, I want to move my arm, or maybe it's a reflex. So the signal comes from the brain. If it's a reflex, it's going to come from the spinal cord. We'll talk about the reflex process later in a different lecture. But this is moved from the brain or the spinal cord using a motor neuron. Neuron is a nerve cell, and since it's for motion, it's a motor neuron. The motor neuron gets to the muscle, but between the muscle and the motor neuron, there's a tiny little space called a synapse. And in order for the signal to be communicated from the neuron to the muscle cell, a neurotransmitter is released. And that neurotransmitter is called acetylcholine. So a neurotransmitter is what the nerve cell releases to go across the synapse for the signal to continue. Kind of like passing a baton in a relay race. So the acetylcholine binds to the muscle cell. This is the general term is an effector cell. So whenever a neurotransmitter or a hormone goes to its target cell for its specific action to be accomplished, the cell that does the action is known as the effector cell, or the cell that has the effect. So in this case, it's the muscle cell. Inside of the muscle cell, you have those sarcoplasmic reticulum, the smooth ER. Inside of that, you have lots of calcium ions, which are then released. And the release of the calcium ions is what triggers the contraction process of those actin moving closer to begin. So all this, we haven't even started moving the muscle yet. So up here we have the end of our motor neuron. That's called the axon of the nerve cell. And this space, this is the synapse right here. And those little blue dots, those are the neurotransmitters. That's the acetylcholine, which is what ACH stands for. So that triggers the signal to be sent down through the cell, and then your sarcoplasmic reticulum release the calcium ions. They are then going to bind to a structure on the actin to allow the myosin to bind to it. So when we look at the actin up close, there is a protein wrapped around it called tropomyosin. And what the tropomyosin does is it blocks this little spot where the myosin head, that purple paddle, binds. And on the tropomyosin, there is something called troponin. And on the troponin, there is a little spot for the calcium to bind. So when the sarcoplasmic reticulum releases the calcium binds to the troponin complex. When the troponin complex has the calcium, that moves this tropomyosin and it opens up the myosin binding sites on the actin. So we haven't even started moving the muscle yet. So brain sends a signal down the nerve signal, the motor neuron, release the acetylcholine. The acetylcholine triggers the release of the calcium ions. The calcium ions bind to the troponin. That causes the tropomyosin to move, and now the myosin can bind. Once the myosin binds, we can start actually moving the muscle. So calcium ions have to bind in order for the muscles to start moving. So once this myosin head binds to the actin filament, then it is going to undergo what's known as a conformational change and cause the actin filament to be pulled together. And one of the things that has to occur for this conformational change, or this change of shape, is the addition of ATP to the myosin. So the ATP is going to come from cell respiration, and which gets its energy from glucose. Glucose is stored in glycogen in the muscle cells, but the ATP, if it's produced in excess, can be stored in creatine phosphate. So when you start exercising, you can release that ATP from the creatine phosphate, and then you can start breaking down glycogen to make more ATP to help you contract your muscles. Okay, so in the process of contraction, we can see, looking at our myosin, we see that we've got an activated myosin down here. So this is kind of like it's ready to grab the actin and then pull it forward in the contraction process. So whenever your muscle is relaxed and not contracting, the myosin is in the ready to go position. And what has happened is an ATP molecule up here has bound to the myosin, and it has broken into a DP, or adenosine diphosphate, and it has attached the third phosphate to the muscle protein of the myosin. Now, whenever you add phosphate to a protein, it changes its shape. That's what's called a conformational change. Or simply you're changing the form, so you're changing its shape. So in this form right here, it is active and it is ready to go. If the signal from the brain is not sent, then the calcium ions haven't been released. The tropomyosin is still covering the myosin binding sites, and this has nothing to bind to. So once the calcium comes in, so we're adding the calcium now, then the myosin can grab onto that myosin binding site, and that causes the ADP and the phosphate to be released. Because the phosphate is now off of the myosin, it undergoes a conformational change again, and it causes it to pull the actin forward. Then more ATP is added. It changes the shape of the myosin again, grabs the actin, and it pulls it forward. So it's a repeating action. So you're ready, it grabs the actin, pulls it forward, gets another ATP, grabs it, pulls it forward, and it's this continuous ratcheting action pulling the actin filaments closer and closer and closer together. And that's what the contraction process is. One of the things that's going to be significant about this is if you don't have ATP to change the shape of the myosin, myosin actually stays attached up here. So if your muscles are running out of energy, they can't relax. So this is one of the things that contributes to muscle cramps, is overtired, overtrained muscles that don't have enough ATP. Other things that are going to cause problems with cramping is if the muscle can't relax, because in order for the muscle to relax, the myosin heads have to be blocked from that binding site. So the calcium actually has to be pumped back into the sarcoplasmic reticulum. Once the calcium ions are removed from the tropomyosin, then the actin filaments slide backwards and the muscle relaxes. The thing about getting the calcium back into your sarcoplasmic reticulum is this is a form of active transport, and it requires ATP. So again, if your muscle is tired, if you don't have enough energy, your body can't pump those calcium ions back into the sarcoplasmic reticulum. The myosin stays attached to the actin, and that muscle can't relax. It stays contracted, and that's another way that you get a muscle cramp. Other things, so you got your tired, overtrained muscles. This is why you need rest days. You can't work out every single day again and again and again. Your body needs time to recover, to build the muscle to make it stronger, and also to build up its ATP stores. You need that ATP to pump the calcium back into the sarcoplasmic reticulum. And you also need salt. This is, or electrolytes. So Powerade, Gatorade, whatever it is, says, electrolytes to replenish whatever. That's just salt. Powerade, Gatorade, that is Kool-Aid with salt in it. You need that salt in order to properly run your nervous system. And when you are contracting your muscle, you also need to send a signal to relax that muscle. And if your nervous system is not working properly because you don't have enough salt, then you can have a cramp from that process. So the main way that you lose salt is through your sweat. And all of these things can contribute to prolonged muscle contraction, which is cramping. There is a very good video in the playlist about muscle contraction, and then there are 2 videos by my best friend Hank Green about muscle contraction and the general functions of muscles in the body. Okay, so as I said before, the actin can act as creating a gel inside of a cell. So these are those gelatin cross-links formed by the actin filaments, and that creates a thicker fluid here. And then here you see that you don't have those cross-links, and so this end is more fluid. So this kind of is like squeezing the end of your toothpaste that pushes the cytoplasm forward, and this allows for a cell to move using what's known as a pseudopod or a false foot. There is a video of an amoeba eating some paramecium in the playlist, but this is just the way that the cell moves. And it's not just amoebas, this is how our white blood cells move around in our body, especially the macrophages. They move around grabbing onto bacteria and viruses and things that shouldn't be in there and eating them. Another thing that the actin filaments contribute to is allowing the cytoplasm to kind of stream and flow around in the cell. This helps the organelles move around. In plant cells, this is important because it allows the chloroplasts to flow and move, allowing them to have more sun exposure to make more food. Video of that in the playlist as well. The last component of the cytoskeleton are the intermediate filaments. These are the proteins, that is keratin. This is the real actual cytoskeleton. This is what gives the specific shape and structure to the cell. And of course, based on its structure, that is going to determine what function it has. It helps hold organelles in place, and it is basically just a bunch of tightly coiled proteins that make them very strong. So, of course, we want them strong because this is what makes up our skin and our hair and the internal structure of our cells. If this starts breaking down, then your cells can break apart. That is part of what happens in apoptosis, or the programmed cell death. But basically, just very quickly, you have an individual unit of keratin, a monomer. They twist together into a dimer, which then two of those make a tetramer. Then those link together, 8 tetramers, and then this rolls up to make a rope of intermediate filaments, and then that makes up the internal framework or foundation of a cell. So lots and lots of proteins twisted, of course a rope, the more twists in it, the stronger it is. So this makes for very strong components. All right, moving to the outside of the cells and what coats the cell membrane, what's beyond that, and what helps hold them together. So the surfaces are what's on outside, and the junctions are what help hold them together. So when we start looking at plants, plants have their cell membrane, and then just on the outside of the cell membrane, they have the cell wall, and they can have multiple layers of the cell walls, giving them different strengths and structures. Other things have cell walls. Plants, we know, have cellulose. Fungi have cell walls made of chitin. And then your bacteria and your prokaryotes have something named peptidoglycan. And that is a protein, that's the peptide part, glycan, that's sugar, so it's a protein-sugar complex making up their cell walls. Plant cell walls, they are built from the outside in, so the oldest cell wall is on the outside, the youngest cell wall is on the inside. The first layer that's actually between two cell walls, so if this is a plant cell and this is a plant cell, then in between them, that's the middle lamella. That's a bunch of long sticky sugars to help glue those together. Then you have the primary wall, which is just made of cellulose. If that's all you have, then you are going to have an herbaceous plant cell. So think about lettuce or spinach leaves. So they're still nice and flexible. So that's herbaceous herbs. And they are still nice and flexible because they only have one cell wall. Those tissues are called parenchyma. You don't need to know that. If you add a secondary wall, so let's add a secondary wall, we see that that is on the inside of the primary wall. Then it's starting to get tougher and stronger. That's going to be called parenchyma, and that makes the cells crunchier like a carrot. And then you can continue to add more secondary walls. So if we add another secondary wall, it's growing inward, and we can see that's going to start pressing on the interior of the cell. If you continue to add those, then that can actually kill the cell. And then you just have these tubes of all of these cell walls, and that's how xylem gets formed. And that's what makes the conducting tissue to haul water up the plants. If we strengthen that with a protein called lignin, then you have what's known as wood. So that means that the major portion of a tree, the wood part, is actually dead. So if you have a giant tree that that's like this big around, the only living portion of that trunk is probably about an inch or two just underneath the bark. And then everything on the inside is dead. If it's like that and we've got lignin and lots of cell walls, then that's called sclerenchyma, and that's stick-like. So these three different tissue types are parenchyma, collenchyma, and sclerenchyma. Three of my favorite biology words that you don't need to know, but they're just really fun to say. All right, if you have living plant cells, like up here when they are just primary walls, those plant cells need to communicate with each other. It's hard to communicate through a wall, so plants actually have holes going right through those walls, and those holes are called plasmodesmata. And plant communication is transferring water, solutes, salts, and chemicals through those cells. And that can include chemicals that are part of the plant's immune response. And one of the chemicals that is known to be part of the plant's immune system is salicylic acid. And you're like, what is salicylic acid? Why is that significant? Well, if you eat salicylic acid, it is going to cure your headache. There are high quantities of salicylic acid in willow bark. And when the pioneers came to America, they noticed the Native Americans chewing on willow bark and willow twigs, and they did that to get rid of their headache. And then later, the salicylic acid was converted to a different form that was easier on the stomach by a chemist, and that is known as acetylsalicylic acid, better known as aspirin. And then he patented it, and his last name was Bayer, so Bayer aspirin. That all comes from willow bark and part of the immune response of a plant against viruses. Extra bit of knowledge, check. All right, so these are the cell walls, these are the plasmodesmata right here, and that allows stuff to flow through between the cells. All right, animal cells, they do not have cell walls, so they don't have that problem of communication as much, but they do still have things to help hold them together. So these junctions, they're either going to simply be there as structural to give strength to the tissues, or they will be there to help facilitate communication. So tight junctions, think of these as binder clips, just pinching cell membranes together. And then you have desmosomes, which have these deep kind of protein roots that go in between the cell membrane, anchoring cells together. And there's going to be some pictures on the next slide. And then you have gap junctions, which are these protein channels that act like little tunnels, just like the plasmodesmata, for stuff to flow between cells. So tight junctions right here, they just kind of clip the cell membranes together. These are the desmosomes with those deep root-like structures for a deeper anchoring to help cells not be ripped apart. And then down here are gap junctions, which are like little tunnels stuff can go through. Just ways that cells are held together in animals. On the outside of the cell membrane, there's a bunch of goopy stuff. And this has many, many different functions. One of those is going to help hold the cells together, just like the middle lamella of the plants. But the extracellular matrix on different types of cells are different. And they are different from different species and person to person. So the extracellular matrix actually makes up the biochemical fingerprint of a cell. So you have things that are glycoproteins, proteoglycans, fibronectins. So these are all just different kinds of proteins with other stuff stuck to them. This is a protein with sugars right here. Fibronectins are fibers that help, interweave and connect everything. Don't really worry about all of that stuff and those names. Just know that there's a bunch of stuff on the surface that helps hold them together. So proteins with sugars, all of this. But the thing that's important about these as these function in cellular communication. So hormones are going to bind to these proteins to trigger a cellular response. This is what the immune system is going to look at to see if this is a self cell or a non-self cell. If the proteins are wrong, meaning it's non-self, then you have an intruder that triggers the immune response, and the immune system tries to kill it. So it's important that these surface proteins in your body are correct, or your immune system may attack it. That's one of the important things about getting a transplant organ is you have to try to match with the donor, because if your surface proteins are very different, then there's a much higher likelihood that the body will reject those organs and attack them as non-self. All right, last but not least in this video, there is a fantastic video called The Inner Life of the Cell in the playlist. This is one of my absolute favorite biology videos. It's a Harvard Medical School animation. It shows all the different organelles in there. It shows a vacuole being dragged across a microtubule. It's my favorite little thing. But the video is showing how a white blood cell, which is moving through the bloodstream, undergoes a change in shape so it can slide out of the bloodstream and start hunting for bacteria inside of the body. It's a great video. Make sure you watch that one, please.